I'm your host, Nick Jikomis, and today I'm speaking with Dr. Bryan Roth. Dr. Roth is a distinguished professor of pharmacology at the University of North Carolina, as well as a member of the National Academy of Medicine and the National Academy of Sciences. His lab studies drugs and the brain. They study how drugs work in the brain. They study how new drugs can be developed to help treat psychiatric illness. And a lot of their work is focused on psychedelics, how psychedelics work in the brain, creating and engineering novel psychedelics with new properties that make them therapeutically useful. And he's one of the leading experts on psychedelic drug action in the world. He's been studying psychedelics since the 1980s, longer than I think just about anyone on the planet. And so I actually talked with him on an early episode of the podcast, and I'm having him back on a couple of years since that episode to talk about some of the latest research in psychedelic science. So we did a little bit of overview of some of the basics. We talked about serotonin 2A receptors and the evidence for why they are largely responsible for the psychedelic effects of psychedelics. We talked about neuroplasticity, how the 5-HT2A receptor may or may not be involved in the neuroplastic effects of psychedelics. We talked about other receptors that various psychedelics interact with. We talked about the TrkB receptor, which binds to an endogenous compound called BDNF, which is an important growth factor in brain development that has to do with neuroplasticity. We talked about various different psychedelics and psychoactive drugs, things like classic psychedelics, so psilocin, DMT, LSD, things like ibogaine, things like ketamine and MDMA. We talked about the differences in terms of the mechanisms by which they work. And we talked about why some of these drugs can have similar effects even though they work very differently. So for example, why do psilocybin and ketamine both have rapid antidepressant effects even though they work in different ways? We talked about various recent results to do with neuroplasticity and psychedelics, and a lot of the details just involved in that stuff and what some of the latest research has said. So if you're interested in psychedelic science, how psychedelics and related psychoactives work in the brain, some of the molecular details around that, and some of the latest findings and what Bryan's take is on those, this will be a great episode for you. As always, if you enjoy the content I'm producing, please like, share, and subscribe. Don't forget to check out my Substack, mindandmatter.substack.com. You can sign up for my free weekly newsletter or become a paid supporter to help the podcast grow and sustain itself. And as always, I appreciate you for tuning in. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food-sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy-free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it with the first meal of each day, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. And there's even evidence indicating that vitamin D deficiency is correlated with more severe cases of COVID-19 in those who get infected. Every time I go into the doctor each year for a checkup, I'm always told that vitamin D deficiency is very common and I should be supplementing on a daily basis. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase. Hey everyone, I want to take a minute to tell you about a really cool health monitoring device I've been using for several weeks. It's called Lumen, and it's a handheld, pocket-sized device with a sleek design. It measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs. It improves your ability to burn fat, which decreases your hunger levels and makes your body less dependent on snacking, and it can increase your energy levels by helping you develop a high-functioning metabolism. I use this device in the morning, before bed, and after meals and workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I need to fast each day to promote fat burning. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. The easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MIND, M-I-N-D, in all capital letters to get $50 off your Lumen device today. And with that, here's my conversation with Dr. Bryan Roth. Does?
Yeah, I'm a professor of pharmacology at University of North Carolina, Chapel Hill School of Medicine, and my lab studies sort of CNS medications and technologies for discovering new medications for serious mental illnesses. I'm also a psychiatrist by training, and I spent many years treating treatment-resistant schizophrenia.
And one major component of what your lab does is study psychedelics and how they work in the brain and things related to that. So how long have you been studying psychedelics in your lab / how long have you been studying this yourself as a scientist?
Since 1983. So after I finished my PhD in St. Louis, I did a postdoc at the National Institute of Mental Health in the late Herminio Costa's lab. And that's where I began my studies on 5-HT2A receptors and psychedelic drugs.
And so, many people...
A long time.
Yeah,
basically.
longer than most.
Too long.
Well, I don't know about too long. So serotonin 2A receptors, these are the so-called psychedelic receptors, as they're often referred to. These are the ones that probably most listeners of this podcast will have some level of familiarity with. But can you give everyone just a very, very basic, short overview of what these receptors are and why they're famous in the context of psychedelics and psychedelic drug action?
Yeah, they're particularly important for psychedelic drugs because I would say there's overwhelming evidence in both preclinical models as well as in humans that the psychedelic actions of classical psychedelics, so these are drugs like psilocybin, mescaline, DMT, LSD, and so on, all mediate their psychedelic actions by activating this receptor in the brain. The best evidence for the involvement of 5-HT2A receptors in psychedelic drug action comes from studies that were done in Franz Vollenweider's lab by Dr. Vollenweider and Dr. Preller over the years. And what they showed is that pre-administration of a 5-HT2A preferring antagonist drug, this drug ketanserin, which in Europe is approved for treating hypertension, but has 5-HT2, pretty strong 5-HT2A blocking activity, that when humans were pre-treated with this and then given, I would say, fairly substantial doses of either LSD or psilocybin, that the psychedelic effects were completely gone. And I have heard sort of anecdotal reports of humans who have taken more selective 5-HT2A antagonists, M100, 907, for example, and basically reported the same thing. Mm-hmm. And in animals, basically all of the quote unquote psychedelic effects can be blocked by pretreatment with a 5-HT2A antagonist, or in mice where the 5-HT2A receptor is genetically removed. Mm-hmm. So I think it's pretty solid that the 5-HT2A receptor is responsible for the psychedelic actions of these drugs.
Mm-hmm. Yeah. So if you give them psychedelics, they have a psychedelic experience. If you give them a drug first that blocks 5-HT2A receptors, gets in the way of the psychedelic interacting with the receptor that basically gets rid of all of the psychedelic effects. And if you do comparable experiments in mice, or you remove the receptor in mice using genetic tools, you get the same basic result. The behavioral effects of the psychedelics that we take as proxies for the hallucinatory effects go away.
Yes.
And can you remind everyone, so in mammals, in the mammalian brain, how widespread are 5-HT2A receptors, and where do we tend to see them in the brain?
So they're most highly concentrated in the cortex, both in mice and in humans, which I would say makes sense because psychedelics alter how people perceive reality. And the receptors themselves are found in a particular type of cell in the cortex called pyramidal neurons. And they appear to be preferentially localized to layer 4 and layer 5 pyramidal neurons in the brain. And within those neurons, they're highly localized to the cell bodies and what are called the apical dendrites. And when psychedelics activate those receptors, they cause the neurons to fire in a very disorganized fashion. And this disorganized firing is thought to be responsible for the psychedelic actions, because these neurons basically integrate information from multiple cortical and subcortical areas to give us the experience of reality that we experience it. And basically what psychedelic drugs do is they inject noise into these neurons, therefore making it apparently more difficult to basically classify internal experiences and external experiences differently, so that people, when they're under the influence of a psychedelic, they believe the experience, basically. Mm-hmm. So this is another characteristic of psychedelic drugs, that they cause an experience which for many people have a very deep meaning for them. We're not at all clear why this occurs, but likely it's through the 5-HT2A receptor.
Mm-hmm. And, normally when we think about receptors, we think about them as being on the outside of the cell, or in the cell membrane, listening right to things coming from the outside. Are these particular receptors, are they only found in the cell membrane, or can they be found inside the cell as well? And what do we know about that?
Well, they're always membrane bound because they're what are called integral membrane proteins, so they can't exist floating around free in the cytoplasm of cells. But we've known for decades now that receptors for psychedelics, as well as receptors for many other drugs, are found both on the cell surface as well as inside the cell. And it varies tremendously from receptor to receptor. Some receptors, I think the delta opiate receptor, for instance, it appears to be primarily intracellular, whereas other receptors appear to be primarily at the cell surface. But basically all receptors in the brain and elsewhere exist in intracellular pools as well as plasma membrane pools. So this is very common. And we've also known for decades that these receptors inside the cell can signal. For historical purposes, this is something I studied in my PhD thesis. I studied opiate receptor subcellular localization, and I found that opiate receptors were found intracellularly. So this was in the early 80s, mm-hmm. basically, and that they were capable of signaling through canonical signaling pathways. So this is a thread that has been around for many, many years, and it was shown, I think, in the 80s there was work by the group at Janssen Pharmaceuticals that did subcellular localization studies of 5-HT2A receptors in rat brain. And what they found was that a significant proportion of the receptors were intracellular. So I think this is, and we in, previously published studies with antibodies that were relatively specific for the 5-HT2A receptor. This was work done in the 90s. We also showed they were intracellular. Some of them were complexed with arrestin, which is a transducer protein, and that various drugs, so drug administration, could change the subcellular distribution of the receptor, both in cell culture as well as in neurons in vivo. So it's, I would say, it's pretty well accepted that these receptors are intracellular as well as plasma membrane bound.
Mm-hmm. And so the ones that are intracellular within the cells, are there endogenous functions that have been worked out there, or are they mostly just sort of sitting there waiting to be deployed to the cell membrane when the appropriate conditions are triggered, or can certain drugs or endogenous compounds actually get inside cells and activate them?
Yeah, usually the way this occurs is that the receptors are at the cell surface. A drug that activates them binds to them. This causes the receptors to be internalized. And then when they're internalized, then they can continue signaling. So typically, this is the way it happens. There are reports of direct activation of intracellular receptors by drugs that can cross the plasma membrane. So this was shown for opiate receptors and beta adrenergic receptors some years ago.
In terms of the endogenous ligands for these receptors, so let's just talk about serotonin 2A receptor and serotonin. Is serotonin mostly or always activating the receptor via the extracellular space outside the cell, or are there instances where it can actually get inside and initiate signaling within the cell?
Well, certainly serotonin itself will cause the receptor to be internalized. And so after the receptor is internalized, if the serotonin stays on the receptor, then there could be continued signaling. I don't know of any data showing that this occurs, but it's certainly theoretically possible. But typically, serotonin is positively charged and would not cross plasma membrane. So in most experiments, if you apply serotonin, it's only going to interact with surface receptors.
Mm-hmm. And so there's good evidence, as you said, that the 5-HT2A receptor is critical for the psychedelic, the psychoactive component of what psychedelics do. So there's that side of it. They have these very remarkable subjective effects that they induce. This receptor is very important for those. There's also the effects on neuroplasticity that psychedelics have, and that's an intense focus of study. And one of the sort of major areas of focus, I think, for many people right now is to address the question of the extent to which the psychedelic effects, the psychoactive effects, can or cannot be dissociated from the neuroplastic effects that these drugs have. So with that in mind, what is the evidence that the 5-HT2A receptor is important for some or most of the neuroplastic effects that psychedelics are associated with?
Yeah, the best evidence for that comes from a series of studies that were done by several groups now, showing that pretreatment with a 5-HT2A antagonist basically blocks the effects of psychedelic drugs on spine formation or dendritic elaboration. So there are now several of those studies in vitro that have shown that by different groups. And then I would say there's mixed data in vivo. So there's a recent paper that was published in Nature by Guy Dolan, suggesting that the plasticity-inducing effects of psychedelic drugs on a measure that they call social plasticity is due to 5-HT2A receptors, because that was blocked with a 5-HT2A antagonist. By contrast, there is a report by Thompson's lab a couple of years ago, maybe 3 years ago now, showing that the enhanced plasticity that's seen by psychedelic drugs in the hippocampus, so a different brain area, is not due to 5-HT2A receptors. And I would just make a little aside at this point that hippocampus doesn't really express 5-HT2A receptors to any extent. Mm. So it would not be surprising that if there's an effect of psychedelic drugs on hippocampal plasticity, it's not due to 5-HT2A receptors. It turns out that drugs like psilocybin and LSD interact with dozens and dozens of different receptors. And so it's quite possible and even likely that in brain areas where there aren't 5-HT2A receptors, that the main effects would be mediated by some other receptor. I would say by contrast, there is one report that was published in Nature Neuroscience earlier this year, suggesting that in both the cortex and the hippocampus, that the effects of psychedelic drugs on plasticity are not mediated by any conventional receptor, but are mediated by direct activation of, or direct modulation of the TrkB receptor. This is a neurotrophin receptor by psychedelic drugs. So there is this other report that would appear to be contradictory to the prior reports of the essential role of serotonin receptors in psychedelic drug actions, both therapeutic and psychedelic.
Mm-hmm. So a couple things to think about when we're thinking about all of these results. One is the difference between in vitro studies and in vivo studies. So it sounds like people have done multiple experiments in vitro, in a petri dish basically, where they give psychedelic drugs and they see neuroplastic changes, they see new spines forming and things like this. If you block the 5-HT2A receptor, you basically block most or all of those effects. And then experiments in vivo, when they're inside of live animals, it sounds like the results are a little bit more mixed. And is that surprising, or is that common where you see you don't necessarily see the in vitro studies lining up with in vivo studies?
Yeah, that's sadly all too common. So neurons in culture, of course, are not neurons. If anything, they're immature neurons. So just to put this in perspective, many, many years ago, I studied the developmental regulation of 5-HT2A receptors in rats. And what I found basically was the receptor really wasn't expressed until around 14 days postnatal. So if you're studying neurons in culture, neurons are usually harvested from days, embryonic day 18 mice. So mice that have not been born. So the neurons are very immature. And what we have found in preliminary studies is that if you quantify the amount of 5-HT2A receptor in cultured neurons, there really isn't any receptor until day 14. And so in these various in vitro studies, it'll be very important going forward to actually make sure that people are studying neurons that actually express the receptors. So that's one sort of complicating factor. The other thing is that there are, I would say, the field of in vitro neuroscience is filled with studies where they see something in neurons in vitro that they don't see in vivo.
Mm-hmm.
And typically this is because of developmental considerations or things like that. So it would be fairly common,
Mm-hmm.
not unsurprising.
Yeah. And so I guess the basic point for people is when people are doing in vitro experiments, those are very easy to do, and they can be much more precisely controlled in the lab in many ways compared to trying to do something in the brain of a live animal. But the cells in that dish are simply in a different context than the cells in an intact brain. There might be at a different developmental stage. There's a different, soup of stuff floating around the brain compared to the petri dish, and you can't assume that the one set of experiments will translate into the other context.
Right. And they frequently don't.
Yeah. And then, the other consideration that you brought up is, different people doing experiments are doing them, in different brain regions, which are going to have different levels of receptor expression and things. Is there strong evidence in pyramidal neurons in the cortex, neurons which do express high levels of 5-HT2A receptors, that in vivo psychedelics are directly inducing plasticity in those neurons?
So nobody has... So there are studies where people have looked at neurons in vivo. This is primarily the work of Alex Kwan. But we don't know if those, and what he shows is that psychedelics enhance plasticity. And it's due in vivo, at least to some extent, to 5-HT2A receptors. But the problem is that the cells that he's looking at probably do not express 5-HT2A receptors. So he uses a Thai-1 reporter mouse. This is sort of a technical thing. And we have made a reporter mouse for the 5-HT2A receptor. So we basically have tagged the receptor so that we can visualize it in living mice. And what we find is that there's very little correspondence between Thai-1 expressing neurons and 5-HT2A neurons in the brain. The Thai-1 neurons are mainly layer 6 neurons, and 5-HT2A receptor expressing neurons are mainly layers 4 and 5. So we actually don't know. My lab is trying to get those studies up and running, but they're technically very challenging to do.
I see. So it sounds like basically people have done the experiment where they can look at, literally through a microscope, look at certain neurons within certain parts of the cerebral cortex. We know that certain neurons in some parts of the cerebral cortex have a lot of 5-HT2A receptors, but it's not all neurons. So they've done the experiment where they give the psychedelic, they're looking at certain neurons and they see plasticity. So they see new spines forming and things like this. But what you're saying is the neurons they're able to look at in those experiments aren't actually the ones that have 5-HT2A receptors. So it's some kind of indirect thing happening.
Yeah. Surprise, sadly.
Yeah. Just unlucky. Okay. So psychedelics have neuroplastic effects. We know that in certain contexts, these can depend directly on 5-HT2A. In other contexts, it's a little bit more ambiguous and things like, and it's just complicated. We haven't fully worked it out yet. On this question of whether or not the psychedelic effects, the subjective effects that they induce can be dissociated from the neuroplastic effects, what do you think the state of the art is there? Has that question been answered yet, or is it still unknown?
Well, certainly it's been answered in vitro because I think David Olson's group has shown that a number of non-psychedelic drugs, including glyceride, can induce plasticity in neurons in vitro. And of course, glyceride is not a psychedelic drug in humans. So there's, pretty strong evidence that at least what you're measuring in cells in culture. We also have reported we and others have reported data in mice with non-psychedelic drugs, So John McCorvey's group reported that bromo LSD, which is not psychedelic in humans, absolutely not, apparently has antidepressant-like actions in mice. And they also showed that this drug Ariadne, which is, I would say, less psychedelic. It's not entirely non-psychedelic, but is a less potent psychedelic, also has some antidepressant-like activity. And we recently posted a paper on bioarchivics showing that lisheride directly has antidepressant activity in mice, antidepressant drug-like activity. So our group published a paper in Nature showing a non-psychedelic compound is ostensibly therapeutic, and other groups have reported the same thing. So there's a lot of data in mice that drugs that interact with the 5-HT2A receptor activated to some degree are not psychedelic, but they have therapeutic drug-like actions in mice. Whether this will ever translate to humans is sort of the big question. So we don't know. There are hints, though, that this is possible because both bromo LSD and lisheride, which are not psychedelic, as well as Ariadne and in sort of anecdotal reports, have all demonstrated potential therapeutic actions in humans. So I think the possibility is there, but we don't really have the best compounds yet to test in humans to test this hypothesis. So a number of us are trying to test this hypothesis directly by basically making compounds that would be suitable for testing in humans.
Mm-hmm. Yeah, and there's lots of considerations for here. So I'll just briefly mention some things for people before I dive into some other questions for you. So neuroplasticity is a pretty broad term. There's many different forms of plasticity and many different mechanisms involved. So it's not just sort of one thing. Different drugs, are going to interact with different things in the brain, and they may or may not induce different forms of neuroplasticity. One of the things I wanted to ask you about related to this general topic is, we know that all kinds of drugs, psychoactive and non-psychoactive, can have neuroplastic effects. SSRIs, almost any, addictive drug that you can think of. But when we talk about the psychedelics and the effects that people have been seeing and studying in them in particular, one of the ways that people have been trying to distinguish them from other drugs is in talking about the nature of the neuroplastic effects they induce. So one of the new terms that's been introduced recently by certain groups is psychoplastogen. And the idea there is apparently that it refers to drugs that can produce rapid and sustained effects on structural plasticity. So the ability of, new spines and things to sprout up or go away. Does that term distinguish psychedelics from other drugs or not?
I don't really like the term that much. If you think about the term psychoplasticity, what it would infer is any compound that's psychoactive induces plasticity induced by any psychoactive compound. And we know all psychoactive compounds induce plasticity. Or if they didn't, you would not be able to remember that you took a drug, basically. So if you drink, if you have a enjoyable drinking experience, that experience is remembered, and the memory occurs by plasticity. That's how we remember things. That's how the brain works. So I prefer the term just neuroplasticity. I don't think there's any data yet that psychedelic induced plasticity is different from that induced by other drugs. We just have to see
Mhm.
I always thought it was sort of a marketing technique, basically.
Well, yeah. In my
You
reading
come
of the
up with a, if you come up with a new term, then, if it captures the imagination of people, then it becomes used. But
yeah, in my reading of the term as it's been used, it seemed like an attempt to distinguish things like classic psychedelics from other kinds of drugs by referring to their ability to induce rapid and sustained structural plasticity. But the thing that confused me about that is there's many other non-psychedelic drugs that we know can induce rapid and sustained plasticity, right?
yeah, cocaine for one, amphetamine. So it was shown by the Rob Malenka's lab decades ago that a single dose of cocaine induces long-lasting, long-term potentiation, which is a type of plasticity, which can be remembered by the mouse for a year, basically.
Mm-hmm.
So it's not, it doesn't quite. I know what they're trying to do, but it doesn't. I sort of object to the term. I'll just say from a philosophical perspective, that ultimately it depends on how people in the sciences use the term. So if people in neuroscience use the term only to discuss psychedelics, then that will be the term, basically. And there may be in the Oxford English Dictionary, there may be some paragraph about a debate about the term, but if people use the term and it's used widely, then it's the term, basically. But I don't like to use the term, basically because it implies something that we don't really know is true yet. It could be true, but we don't have any data for it.
And one of the things I'm hoping you can unpack for people a little bit is, so for non-scientists listening that don't have a background in this, it's a little abstract when we talk about, drug goes into the brain, it binds a receptor, and then plasticity happens.
Right.
What's happening in between? So a drug, let's just take psilocybin or LSD as an example, a classic psychedelic. It gets into the brain, it binds to serotonin 2A receptors and other receptors. And then somewhere after that, plasticity happens, new connections sprout up and things like that. What's happening in between that connects those dots?
So my lab with Peter Penzis investigated this in some detail now 14 years ago. And what we found was that synaptic scaffolding proteins and protein kinases that have historically been involved in new spine formation appear to be involved in that process, and that it was due to the 5-HT2A receptor. So there were synaptic scaffolding proteins called MUP1, technically a PDZ binding domain protein, which we know interact with the receptor. And then various protein kinases downstream of that are activated, ultimately leading to some cytoskeletal rearrangement. And in our paper, which I urge people to read, but no one reads because I don't think anybody reads anything that's more than 5 years old. We show fairly conclusively the involvement of specific kinases and other proteins in this process. That's for the initial phase of plasticity. So we were looking at, in our paper, what we found was that psychedelic drugs can induce spine formation, which within 30 minutes of exposure, which is very fast. In terms of the maintenance of spines, it's likely that other pathways involving BDNF and TrkB receptors are involved. So it's sort of well known in neurosciences that BDNF and TrkB that pathway is responsible for the maintenance of new spines and synaptic plasticity. And there's a fair amount of data now that psychedelic drugs can enhance the expression of BDNF, and that spine formation is dependent on TrkB receptors. That's very similar to plasticity that's induced basically by any drug, including ketamine. So ketamine is another, rapidly acting antidepressant drug. It's pretty well accepted that activation of BDNF and TrkB is essential for the plasticity inducing effects and the antidepressant effects of ketamine, for instance. So it wouldn't be surprising if this system is also essential for psychedelic drug actions.
And does, when plasticity is induced by a drug or an endogenous compound, does that necessarily have to involve changes in gene expression, signals that go all the way to the nucleus and turn genes on or off?
Not necessarily. So there is local protein synthesis that occurs in the spine. One of the people that just won the Brain Prize basically discovered this process. So underneath the spines, it turns out that there's messenger RNA and rough ER and so on. And there can be local protein synthesis that occurs. So it doesn't necessarily have to go, the signal doesn't necessarily have to go back to the nucleus and alter gene transcription for the neuron, at least initially. Long term, of course, there are going to be changes in protein translation and transcription. But how these changes ultimately are responsible for the plasticity that may or may not occur with psychedelics is not known.
And, another concept I wanted to ask you about that's relevant to some recent findings is, so when we talk about neuroplasticity, that's the actual like physical changes that happen per se. So a new spine, a new structure, a new synapse forms. There's also the concept of metaplasticity. What is that?
I have no idea because I don't study it.
I see. So the recent paper from Gould Dolan's lab that looked at social reward learning was looking at and distinguishing plasticity.
So I just looked it up in Wikipedia, and it refers to the plasticity of synaptic plasticity.
So yeah, I think people would say it's the extent to which plasticity can happen, or how easily a new synapse can form, or something like that. One thing that I was hoping to ask you about related to that was so when we talk about synapses forming, the connection between two neurons, we talked a little bit about the intracellular side, some of the proteins and kinases that are involved in synapse or spine formation. What about the extracellular space and sort of the physical structure of that? How much relevance is there to the extracellular composition in terms of whether or not new synapses can form?
Yeah, well, it's key, of course. When a synapse forms, it has to meet the adjacent neuron, basically. And to do that, it has to, it's guided across by the extracellular matrix. So extracellular matrix proteins are essential for all types of plasticity, ultimately, or involved in all types of plasticity.
I see. So not only does the inside of the neuron needs to change, but it needs to be able to, essentially physically move across a scaffolding, a physical substrate, in order to move around things. Right.
And there's recognition by basically the presynaptic membrane and the postsynaptic membrane. So there are adhesion proteins that cross the synapse. And these were first discovered by Tom Sudoff, and he got the Nobel Prize for that some years ago. So basically, it's a fairly complicated, as you can imagine, it's like everything else. It's a fairly complicated situation, which is regulated at multiple levels. So there's extracellular matrix, there are adhesion proteins, there are signals, diffusible signals that go both ways. So there are lots and lots of processes that are involved, including, extracellular matrix proteins.
Another thing that's interesting, I think, in this general field is, you've got results on things like rapid antidepressant effects from different kinds of drugs that work in different ways. So something like psilocybin, that's a classic psychedelic. It binds the 5-HT2A receptor and other receptors. Then you got something like ketamine, which is a dissociative anesthetic, and it's operating at least in part through NMDA receptors. But it also has these rapid antidepressant effects. So how do you start to think about this, where you kind of have the same endpoint being measured, the same effect, but it's coming from drugs that have, at least that would appear to have very different mechanisms of action?
Yeah, I think ultimately, if we focus on the glutamatergic neuron, so 5-HT2A receptors are on pyramidal neurons. These are glutamate-expressing neurons, and one of the peculiar things about ketamine. So ketamine is an NMDA receptor antagonist, and what it does at sort of sub-dissociative levels of administration is it preferentially interacts with NMDA receptors on GABA neurons. So GABA neurons are inhibitory, and it blocks the excitatory input to the GABA neurons. So it's basically blocking inhibition. And what that does is that relieves a inhibitory brake on pyramidal neuron firing and promotes increased firing of pyramidal neurons, basically, okay? And psychedelics do the same thing. Except they do it directly. And we have recently found, using we haven't published this, but I presented it at meetings, that NMDA receptors, the receptors for ketamine, appear to be in close proximity to 5-HT2A receptors in those neurons. So there could actually be a direct physical link. And it was previously shown by Rex Wang's lab in the 90s that psychedelics alter NMDA currents. So I think we're getting sort of converging evidence that it's basically a final common pathway related to firing of pyramidal neurons. Ketamine does it sort of indirectly, and psychedelics do it directly.
I see.
And that might, it could be in part why psychedelics apparently have a longer duration of action than ketamine, at least clinically.
I see. So ultimately, when we're thinking about effects in the brain, we're talking about changing patterns of activity of neurons, and there's more than one way to do that. If you want to, increase the amount of excitation in a circuit, you can increase the activity coming from the excitatory neurons. You can also decrease the level of inhibition going on to them from the inhibitory interneurons.
Right.
I see.
Yeah, you can do it directly or indirectly, basically.
And
the data that we have and others have suggests that ketamine does this indirectly, and classical psychedelics do it directly.
Mm-hmm. And is it common for a drug to have dose-dependent effects like that, where, at a low dose, it's interacting preferentially with, one cell type, and then at a higher dose, it starts interacting with others?
It's not uncommon. Yeah.
And how does that work? Do different cells have like different receptors with different levels of affinity for the drug, or why does that happen?
So I don't know why that is, but it's a phenomenon that I think was initially described by beta-mogadam maybe 15 or 20 years ago, and has been, recently replicated. So it's a, you would probably have to ask Beta. She's sort of the world's expert on this, why it is that low doses of ketamine particularly target interneurons and, inhibitory interneurons, and higher doses interact with NMDA receptors on excitatory neurons. So I don't know why that is, but it's something that's, been described multiple times in the literature. So it's a real phenomenon.
Mm-hmm. And then, another drug worth talking about in a similar vein. So you've got something like a classic psychedelic, which we've been talking about, where we're thinking about 5-HT2A receptors, ketamine, a NMDA receptor antagonist, and then you've got something like MDMA. So how does MDMA basically work? And do you think of MDMA as a psychedelic, or what type of drug is it? / how should people think about classifying drugs in terms of their mechanisms?
Yeah, so NMD or MDMA is, ordinarily you would put it in the bin of psychostimulant because it has amphetamine-like actions. So it causes the release of dopamine and serotonin. That's how it works. It does it by interacting with transporters for dopamine and serotonin. And MDMA also has, I would say, low affinity for 5-HT2A receptors. It has a metabolite, MDA, which has higher affinity for 5-HT2A receptors, but they're still sort of weak. Because MDMA primarily causes the release of serotonin and dopamine, there's data in the literature that drugs that block dopamine and serotonin receptors can block certain effects of MDMA. So some effects of MDMA are blocked by 5-HT2A receptor antagonists, some are not. I think 5-HT1A receptor antagonists may block some effects. And the locomotor activity of MDMA can be blocked by dopamine antagonists. So it has sort of a complicated mechanism of action. It's not entirely clear how this leads to the potential therapeutic actions of MDMA. MDMA also interacts with a family of receptors called traceamine receptors, which are found in the brain, and it's possible that interactions with traceamine receptors, psychedelics also interact with traceamine receptors to some extent. It's conceivable that actions at traceamine receptors are responsible for the therapeutic actions of these drugs, but we just don't have any data on that. So we consider, I sort of consider MDMA as just a psychostimulant.
Mm-hmm.
I think people that study MDMA refer to it as a entactogen or empathogen because it increases one's empathic processes. But it's not a psychedelic because as far as I understand, it doesn't induce hallucinations, right? I haven't heard that.
Yeah, I don't think anyone. I've never heard that.
Yeah, so psychedelics induce hallucinations, right?
Mm-hmm.
As a minimum, and MDMA doesn't. So ordinarily, we would not classify it as a psychedelic. Again, I think it's sort of this branding. If people can brand MDMA as a psychedelic, it makes it sound more sexy, I think.
Mm-hmm. Yeah, I mean, it's
in the past. So in the past, by like 5 years ago, when no one was studying psychedelics, other than my lab and 2 other labs in the world, none of us considered MDMA a psychedelic. But now that there's this huge universe of people that are interested in these compounds, they're sort of binning them all together.
Yeah.
And again, if ultimately, people decide that any drug that causes you to feel strange is a psychedelic, then that's what they'll be, basically.
Mm-hmm.
I sort of have been advocating a pharmacologic classification of these drugs, basically because that's the way we in pharmacology, that's how we define drugs, is
Mm-hmm.
based on their mechanism of action. So, but again, we'll have to wait for 10 years to see how the word psychedelic is actually being used.
Yeah. I mean, ultimately, you're describing a linguistic phenomenon, which is not specific to this field by any means. But yeah,
Right.
it sort of seems like the word psychedelic, in my view, and of the read of the last few years of usage, it's almost evolving just to mean something like rapid acting next generation psychiatric medicine or something.
Yeah, I don't know if people that went to the psychedelic science conference would use that definition. I think, my sense is the people that are sort of in quote unquote the psychedelic community would consider ketamine, ibogaine, salvia, classical psychedelics, MDMA, dextromethorphan, et cetera, would consider all of these drugs psychedelic, quote unquote psychedelic. So they're sort of co-opting. So we used to have this term psychoactive
Mm-hmm.
to describe those drugs. So everybody would agree they're psychoactive. And it ultimately will depend. It's sort of like, say 20 years ago, if you wanted to refer to somebody as having a good sense of empathy, you would say they're empathic, right?
Mm-hmm.
And then about 10 years ago, people started using the term empathetic, which was not the preferred term, but it now is. Mm-hmm. Basically, if you go to the dictionary now, empathetic, which was a unpreferred term, is now the preferred term. Okay. And it may be as we go forward that instead of using the term psychoactive, we'll use the term psychedelic.
Mm.
And then I would recommend that we refer to drugs like LSD, mescaline, and psilocybin, DMT as classical psychedelics. And the others as atypical psychedelics. This is the approach that the European Commission on Drugs has taken. So they're sort of the European equivalent to the FDA, the Food and Drug Administration. So they define classical psychedelics and then other psychedelics.
Mm-hmm.
Okay.
So classical psychedelics would be 5-HT2A, like serotonergic psychedelics like LSD, psilocybin, right. DMT. Yeah. And then you've got the other ones. So like salvia acts through a completely different receptor.
Yeah.
And so on and so forth.
Ibogaine, same thing. Yeah. Yeah.
Another interesting question here is related to the duration of the subjective effects of these things. Something like ibogaine lasts a very, very long time. LSD also lasts a long time, not quite as long. But even within the classic serotonergic 5-HT2A receptor activating psychedelics like, say, psilocin, LSD, DMT, there's a lot of difference between them in terms of how long they last. And how does that actually work? Does it just have to do with how long they are sitting at the receptor?
So to a great extent, it's due to what's called pharmacokinetics, basically how fast they're cleared from the body. So DMT is cleared very quickly. It's degraded by monoamine oxidase. Psilocin slower, and LSD's hardly at all. So that's a main driver. Another driver for LSD in particular is that when it binds to the receptor, it binds in what's called a pseudo irreversible fashion. So once it's bound to the receptor, it doesn't come off for like 8 hours. And that's because a lid comes over the top of LSD and basically locks it into place. We don't see that with other psychedelics. It appears to be peculiar to just LSD-like compounds, which have this long duration of action. So we think it's pharmacokinetics as well as the fact that it's when it's on the receptor, it doesn't come off. Drug like DMT has fairly low affinity, so it's gonna come on and off the receptor in the second, within a few seconds, basically, so that as it's metabolized, it basically is completely eliminated. And this is not the case for LSD.
And so you've got these different durations of action. You've got human observations where, some of the clinical results with things like psilocybin show a correlation between the reported intensity of the subjective effects and the actual duration of the therapeutic outcome. That was also the subject of that recent paper about critical periods in social reward learning, where they looked at various drugs, things like psilocybin and LSD, classic psychedelics, as well as things like ketamine and ibogaine and MDMA. And they found this interesting relationship with respect to this social reward learning model they used, where basically the length of the subjective effects was proportional to the duration of time of the drugs being able to open this critical period for social reward learning. And this relates to this question of the efficacy of the potential therapeutic efficacy of the subjective effects themselves. What do you think about that recent result, and what's it going to look like when we have more definitive answer to this question of the therapeutic relevance of the subjective effects?
Well, I would say that what they report is that the duration of action of the drug is correlated with the duration of the effect on social learning. And I would say that's unsurprising. If a drug has a longer duration of action, it's going to have a longer duration of action, basically. So that result was not particularly surprising to me. It was interesting. I think we really need, this is a situation where we really need more human data. So we only really have human data for a couple of well-controlled phase 2 trials for psilocybin, basically. And in one of the most recent ones, it appeared that the therapeutic effect may have tended to wane actually at the end of the study. I think this was the one that was in the New England Journal of Medicine, psilocybin versus S-ketamine. So I think right now what we need are adequately powered, adequately controlled human trials to see really what the duration of action it really is. And I would just, at this stage of the game, I would be agnostic, because we just don't have any data on that. But I would say that the findings in the paper are not unsurprising. That the pharmacokinetics of the drug appear to be correlated with the duration of action. That's sort of classic pharmacology dogma. Mm-hmm.
So another thing I wanted to ask you about related to recent results has to do with, so we've talked about 5-HT2A receptors, serotonin 2A receptors with respect to the classic psychedelics. They're very important. They seem to be essential for the subjective effects. They're likely also playing some role in the neuroplasticity, although the in vivo evidence is more ambiguous there. But as you mentioned, all of these compounds, LSD, psilocin, DMT, basically all of these things bind to a bunch of different receptors in the brain, and each one has a different pattern of affinity for a different set of receptors. One of the ones that you mentioned earlier was this thing called TrkB, and it's bound by BDNF. Can you give people a little bit more background on what those things are and sort of a high level overview of what they're famous for in terms of development and plasticity?
Yeah, these are what are called growth. They're in the growth factor receptor family. So these were originally growth factor receptors were originally identified by Rita Levi-Maltolchini, and there was somebody else, but she got the Nobel Prize for basically discovering nerve growth factor and the various receptors. And BDNF is just brain-derived neurotrophic factor. It's one of dozens of growth factors, and then there are dozens of growth factor receptors, and many of them have been demonstrated to be very important for normal growth and maintenance of the brain and its connections. TrkB has, since I would say Ron Duman's work, in the late Ron Duman's work in the 90s, has been a sort of achieved central importance in the regulation of the effects of a number of drugs, so that induce plasticity. So drugs like cocaine and morphine, which cause plasticity of a deleterious type. So the type of plasticity they induce is dependence. This is all dependent on TrkB. This is well described. Cocaine, amphetamines, morphine. Basically, their long-term effects are all mediated by TrkB. And there's data, compelling data, that the long-term effects of classical antidepressant drugs also ultimately are mediated by TrkB. Okay. So there's data going back a couple of decades that a final common pathway of the action of many drugs that induce long-term changes in the brain is due to activation of the TrkB receptor, directly or indirectly. Okay. So it's another one of these proteins it's well known. If you go to PubMed, there maybe 50 or 60,000 papers on TrkB and BDNF. It's maybe 100,000 papers on BDNF, basically. So BDNF has been, and TrkB, they've been implicated in every sort of synaptic event almost in the brain. Mm-hmm. And as I said, there's data now from a couple of groups that at least that TrkB and BDNF may be part of the final common pathway of psychedelic drug action.
And there was a recent paper that I looked at, but I didn't scrutinize it in too much detail. But it had to do with psychedelics binding to this TrkB receptor, which is what BDNF binds to, and apparently they directly bind to it. So was that known already? And is this paper contradicting the idea that the neuroplastic effects, at least some of them, are coming through 5-HT2A receptors, or could it be both, or is one thing upstream of the other?
So I would say it doesn't. So what it shows is that LSD and psilocin, which is the active metabolite of psilocybin, can bind to the TrkB receptor with high affinity. Mm-hmm. And with the same affinity that they bind to serotonin receptors, and that they can potentiate the effects of BDNF on activating TrkB. And then they show that TrkB is essential for the neuroplastic effects of psychedelics. So I would say the part of the paper where they say TrkB is important for psychedelic action, I think most people would probably agree with that. I don't think that's a particularly unsurprising result. The thing that's a little bit surprising is that they find that that LSD binds with such high affinity to TrkB. It's like 1 nanomolar potency. That's very high affinity. Um, and it's never been reported before. I don't know if anybody's ever looked very closely. There would have been no reason to look. So we're waiting to see if other people see the same thing, basically. So it's a pretty interesting result. It could be true, but we have to see. My lab is, we're working very hard now to see if we can reproduce those findings, because obviously, if they were true, then that would be another potential target that we could utilize for drug discovery purposes. That being said, there are some downsides to directly hitting TrkB. So there were studies that have been done in the past where people have tried to use BDNF as a therapeutic agent for various neurodegenerative diseases. It basically didn't work, and there were a lot of bad side effects. So we think that just sort of willy-nilly activating TrkB in the brain is probably not a good thing long term. But since psychedelics are only taken occasionally, if this is true, it could be, something really interesting. We just have to see.
And is BDNF, is that something that gets sort of released widely across the brain, or is that something that operates in a very sort of specific synapse by synapse fashion?
Yeah, so it's released, so it'll have local effects, but it's typically only released from neurons that are, that activate its release, basically. So it's not normally sort of activated everywhere in the brain. It's found in every neuron in the brain, basically. So typically there's some pathway specificity about the effects of BDNF.
Mm-hmm. And, when we think about drugs like this, or really any drug, whether it's a psychedelic or an SSRI, anything that we want to use to treat treat a psychiatric condition, presumably when someone has, depression or PTSD or whatever, there are certain synapses in the brain, certain circuits that you want to be more plastic, and, create new connections or take away some connections. And there's presumably many that you actually don't want to change. But when we administer drugs to people, the drug goes through the bloodstream and it sort of gets in the brain. It basically goes everywhere. And so how do you, as someone who's sort of a molecular mechanistic laboratory scientist, but also has training in psychiatry, how do you start to think about this, this issue of, we want to have very, very targeted changes that we induce in the brain to help fix it. And yet our drugs, basically go everywhere.
Yeah. Well, psychedelics go everywhere, but their receptors are not everywhere.
Mm.
So one way of addressing sort of the question is to make psychedelic drugs that only hit the 5-HT2A receptor and see if they're
even more selective.
Yeah. So we have those compounds now. And they are therapeutic, at least in mice. So that's one way. Another sort of futuristic way is to use this technology that my lab invented called chemogenetics. And what chemogenetics allows us to do is to target distinct neuronal populations and turn them on and turn them off with a pill, basically. Somewhat akin to optogenetics, except you don't have to put an optical fiber in somebody's brain and turn it on.
And I think most listeners probably haven't heard of chemogenetics or DREADS or some of this cool technology that you invented. Can you walk people through how that works at a high level?
Yeah, I have 5 minutes left here. So
okay, yeah, we'll just end it there.
Yeah, so chemogenetics basically use receptors like serotonin receptors that have been engineered so that they are activated only by a drug which is inert. And so these can be put into any neuron in the brain, and then they can, by administering of an inert drug, we can turn the neurons on and turn them off, basically in a highly robust fashion. And I would say in the neurosciences, the technology is used sort of ubiquitously. So if you go to the neuroscience meeting, there will be literally hundreds and hundreds of posters using this technology. And it's also it's now been shown in multiple papers to be very effective in non-human primates, unlike other technologies like optogenetics, which are very challenging when you get to bigger brains. So we think that ultimately this, another approach would be focused. There are focused ultrasound procedures now that are being developed to target particular circuits in the brain, and there's focused transmagnetic cranial stimulation as well. These are non-invasive technologies to modulate the activity of particular neurons. So that's the approach we're taking, is to make drugs that are way more selective for those neurons, and then also to develop technologies where we can, once we find the neurons we want to modulate, we can turn them on and turn them off at will.
I see. So one strategy is tweaking drugs so they're binding fewer types of receptors and they have more specific activity.
In this case, only one, right? Only the 5-HT2A, which is only found in certain neurons in the brain, right?
What's the name of that drug?
ZX something. They all have code names. We have a ton of them basically now, and they all seem to work just fine in mice. So at least in mice, it appears to be the 5-HT2A receptor. We're pretty clear about that now.
All right. Well, Professor Bryan Roth, I know that you got to go. You've got stuff to do, but thank you for joining me again. This was fascinating, and have a good day.
Bye.